Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, let's unpack this. Imagine facing a serious health condition, right? And then you find out that even like a common infection could suddenly become life-threatening.
Yeah.
That's the reality for many people living with mitochondrial diseases. And, what if the very powerhouses of their cells are somehow triggering this danger?
It's a really significant problem. For a long time, this increased vulnerability, the susceptibility to severe infections, it's been a bit of a mystery.
Right. We know about the primary issues with faulty mitochondria, the whole energy production thing.
Sure, the cell's power plants.
Exactly. But this infection angle, it adds this whole other layer of complexity and, frankly, danger.
It absolutely does. It's a major challenge in managing these conditions.
So in this deep dive, we're going to explore some really interesting new research. It's starting to unravel why people with mitochondrial diseases seem so prone to these really bad infections.
We'll be looking at the mechanisms, the why behind it all.
And importantly, what this might mean for new treatments down the line.
Yeah, exactly. We're mainly drawing from a scientific paper in Nature Communications and also a health news piece from the Jackson Laboratory covering this work.
Our mission really is to help you understand why a seemingly simple infection can escalate so badly for these individuals.
And what these discoveries could mean for future therapies, it's potentially huge.
Okay, let's get into it. The study we're focusing on spent a lot of time looking at something called polymerase gamma disease, sometimes called POLIG disease. What do we need to know about that specific condition?
Right, so polymerase gamma disease, or POLIG as it's often shortened, is a rare condition. It's progressive and it affects the mitochondria.
Okay.
It stems from mutations in the polymerase gamma gene itself. And this gene is really crucial for maintaining the DNA inside the mitochondria.
So that gene's faulty.
Then you get problems. It leads to a whole range of severe health issues, muscles, nervous system, heart. It's a really debilitating condition.
The study materials also mention the involvement of the Polg Foundation and even Prince Frederick de Nassau. That really adds a human element, doesn't it?
It absolutely does. The Polg Foundation, it was actually set up by the family of Prince Frederick de Nassau. He tragically passed away from complications of this disease.
Oh, wow.
Yeah, so their dedication really highlights just how urgent the need is for better understanding and treatments.
Definitely. Now, to figure out how this all works, the researchers at the Jackson Laboratory, Professor Philip West's team, they used a specific method. They studied mice. Right. Mice with the same genetic mutation seen in human patients.
Yes, that's right.
Why is that kind of mouse model so important for this research?
Well, creating these animal models that, closely mimic human diseases is just fundamental. It's how we make progress.
Right.
This particular mouse model with that specific polymerase gamma mutation let the researchers watch how the disease unfolded in a living system. They could study how the defect impacts different biological processes, especially that critical immune response to infection.
Okay, so we usually think of mitochondria as just, the energy factories.
The powerhouses, yeah.
But Professor West's earlier work suggested they do more, another important job.
That's correct. Yeah, while the energy role is vital, his previous research showed that mitochondria are also key regulators of our immune system.
Oh, okay. So not just energy, they're involved in defense, too.
Exactly. They play a really important part in how our bodies react to threats like infections.
So with that immune role in mind, the researchers took these mice, the ones with the faulty polymerase gamma gene, and introduced a bacterium, Pseudomonas aeruginosa. Why that specific bug?
Pseudomonas aeruginosa is what we call an opportunistic bacterium. It's a major threat, especially for people with weakened immune systems.
Okay.
It causes serious lung and skin infections, and the tricky thing is it's everywhere in the environment. Plus, it can become antibiotic resistant quite quickly, and it causes recurring infections.
So a real problem for someone already vulnerable.
Exactly. For someone with a mitochondrial disease, this bacterium can be incredibly dangerous.
And what happened? What did they find when these specific mice got infected?
This is where it gets really fascinating and kind of counterintuitive. Instead of just fighting the infection effectively, the immune system in these Polgi mice, it went overboard.
Overboard how?
It launched this massive inflammatory response, way over the top. And this overreaction actually caused significant damage to the mice's own tissues, especially in the lungs.
So the defense system hurt the host.
Precisely. It's like the body's defenses went into overdrive and ended up causing more harm than the actual infection in some ways.
Professor West made a comparison to severe COVID-19 cases, didn't he? Where the immune system's overreaction caused a lot of the damage.
Yes, that's a really helpful comparison. In both scenarios, you see the body trying to fight something off, but the immune response becomes uncontrolled, leading to this harmful cascade of inflammation.
So what's the root cause? What's happening at the molecular level in these Polge mice to make the immune system react so extremely?
Okay, so they dug into that. The core issue seems to be the damaged mitochondria themselves.
The faulty powerhouses again.
Exactly. In the cells with this Polge mutation, the dysfunctional mitochondria were churning out high levels of an inflammatory signal called type 1 interferon.
Interferon, okay.
And crucially, they were doing this even when there were no bacteria around. So the system was already on high alert.
Pre-activated almost.
Yes. And this constant interferon signaling then pre-activated a specific bacterial sensor called Caspase 11. It essentially put the immune system on a hair trigger.
Wow. So Caspase 11 is normally supposed to help detect bacteria. Right.
That's its job, yes, to help recognize and respond.
But here, because it's already sort of switched on.
Precisely. In these Polge mice, the overabundance of this sensor, thanks to the constant interferon, meant that even a small bacterial trigger could set off this massive damaging response. It's primed for an explosion.
And this had bad consequences for the immune cells themselves, too.
It did. They observed that immune cells from these Polge mice were self-destructing more often. It's a process called pyroptosis, fiery death, essentially. And when these cells die this way, they release harmful molecules that just fuel the fire, making the inflammation even worse. It creates this really damaging cycle.
And you mentioned earlier there was another study suggesting this isn't just about Polge disease.
That's right. Another piece of research indicated this pattern, this immune overreaction tied to mitochondrial issues, might extend to other mitochondrial disorders too. So it could be a broader mechanism.
This really paints a clearer picture of why infections are such a threat. So, okay, the big question. What does this mean for treatments? Where does this research point us?
Well, this is the really hopeful part. Pinpointing Caspase 11 as a key driver of this hyperinflammation opens up a really exciting possibility.
Which is?
Targeted treatments. Instead of using drugs that just broadly suppress the entire immune system, which has its own risks, obviously.
Yeah, leaves you vulnerable elsewhere.
Exactly. Researchers can now think about developing drugs that specifically block just that Caspase 11 pathway.
Ah, so a much more focused approach. Like a sniper rifle instead of a shotgun.
That's a good analogy, yeah. The big advantage is you could potentially block that harmful overreaction without completely shutting down the immune system's normal ability to fight infections.
That would be huge for these patients.
It really could be. It offers a way to protect against those severe infection complications while letting the immune system still do its basic job when needed.
And you mentioned earlier this might have relevance beyond mitochondrial diseases too.
Potentially, yes. We know mitochondrial damage is linked to other things, certain neurodegenerative diseases, even severe COVID-19, as we discussed.
Right.
So understanding this connection between faulty mitochondria and runaway inflammation could maybe lead to wider therapeutic uses down the road.
The study also talked about something called guanylate-binding proteins, or GBPs. How do they fit in?
Right, the GBPs. So their production gets ramped up by that type 1 interferon we talked about.
All right.
And in the immune cells, specifically macrophages from these Polge mice, the higher interferon meant more GBPs. And it seems these GBPs make Caspase 11 even better at sensing a bacterial component called lipopolysaccharide, or LPS.
So they sort of enhance the trigger sensitivity.
Exactly. They amplify that Caspase 11 sensing, contributing further to the over-the-top inflammation and that cell death process, pyroptosis.
And one more piece, interleukin-1 alpha signaling. What was that about?
Yeah, so when Caspase 11 gets activated in these mice, it also leads to the release of another signaling molecule, a danger signal called interleukin-1 alpha.
Another signal.
Yes, and this one acts like an amplifier. It further activates other parts of the immune system, essentially creating a feedback loop that just makes the whole inflammatory response even worse.
Gotcha. It really is a complex and, like you said, almost self-destructive cycle happening.
It is, unfortunately.
Now, these findings are clearly really important, but like all research, there are limitations. The researchers pointed some out, right?
Yes, absolutely. It's always crucial to remember that mouse models, while incredibly useful, aren't perfect copies of human disease.
They're models, not the real thing.
Exactly. So this specific pulgy mouse model, for instance, doesn't perfectly match any single human mitochondrial disease, even though it shares many features.
And the study did focus mainly on polymerase gamma disease itself.
It did. That was the primary focus. And while the results strongly suggest similar things might happen in other mitochondrial diseases, we definitely need more research to confirm how widely these findings apply across that whole spectrum of disorders.
And what about using lab dishes versus whole animals, the in vitro versus in vivo studies? Any limitations there?
Well, they both have pros and cons. The studies on cells in the lab, the in vitro work, gave really detailed insights into the specific molecules involved, great for mechanics.
Okay.
But the studies in the living mice, the in vivo work, give you a more complex, realistic picture. But that complexity also means more variables, making it sometimes harder to pin down exact cause and effect.
Sure, makes sense. Now, to really underline how significant this is, the researchers themselves commented. What did Professor Philip West, the senior author, say?
Professor West emphasized that their work sheds really important new light on why infections can become so severe for some people with mitochondrial disease.
Okay.
And he also highlighted the exciting part, identifying specific targets, like Caspase 11, that could lead to desperately needed new drugs for these patients.
Finding those molecular targets is key.
It really is.
And the lead author, Jordan Van Poortvliet, what was her take?
She talked about the potential of being able to sort of dial down that hyperactive immune response without just shutting everything off completely.
The targeted approach again.
Right. She described that possibility as a potential game changer for treating infections in these individuals.
A game changer. That's strong wording. It's also worth noting this wasn't just one lab working in isolation, was it?
No, absolutely not. It was a collaborative effort. Involved Professor Steve Murray and Peter McGuire, and of course the vital input and support from the Polk Foundation. It really shows how tackling these complex medical problems needs that kind of multifaceted approach, bringing different experts and perspectives together.
Definitely. Well, this deep dive has really shone a light on such a critical area. It really makes you think. What's a final thought or maybe a question you'd like to leave our listener with?
I think this research really pushes us to consider how understanding this deep link between mitochondrial health and our immune system could truly revolutionize treatments.
Yeah.
And not just for rare diseases like Polja, but maybe for more common conditions where inflammation is a big factor. It makes you wonder, doesn't it? What other health problems that seem unrelated might actually have roots deep down in these fundamental cellular processes?
That's a really powerful thought to mull over if someone listening wants to dig even deeper into this specific research.
Sure. The original paper is in Nature Communications. You could search for that. Or look up the DOI, the Digital Object Identifier. It's 10.1038 STWA 41467 025 599078. And of course, learning more about mitochondrial diseases generally and the work of the Polk Foundation is always valuable. Their websites are good resources.
It definitely feels like a field where we might see some significant progress.
I think so. This research feels like a really meaningful step forward. It offers a real sense of hope, I think, for people living with these conditions.
Thank you for joining us for this deep dive into this crucial research.
Yeah, thanks for having me. The main point to take away is that, yes, damaged mitochondria can trigger this dangerous immune overreaction. But crucially, this new work points towards promising, targeted ways to potentially treat it.
We really appreciate you diving deep at this today. If this sparked any thoughts or questions for you listening, please do share them.
And please join us next time as we delve into another fascinating area of research.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.